I'm Daniel Levine, and this is the Bio Report. The idea of developing cancer vaccines that harness dendritic cells is not new, but Diakonos Oncology thinks it's been able to solve the lack of efficacy that has plagued this approach. The company combines its vaccines with RNA and proteins from a patient's tumor to trigger a robust response by tricking the immune system into recognizing cancer cells as being virally infected. We spoke to Jay Hartenbach, Chief Operating Officer of Diakonos Oncology, about its dendritic cell cancer vaccine technology, how it works, and why its lead indication is an aggressive form of brain cancer. Jay, thanks for joining us.
Hi, Danny. Good to be on. I'm really excited to have the conversation today.
We're going to talk about dendritic cells, cancer, and Diakonos Oncology's efforts to develop a dendritic cell vaccine to combat difficult-to-treat cancers. Let's start with dendritic cells. Can you explain what these are and the role they play within the immune system?
Sure. Yeah, as you've already mentioned, dendritic cells are immune cells that are generally found throughout the tissues, though in an immature form they can be found in the blood after they've left the bone marrow. And the role that a dendritic cell plays for the immune system is to really lie in wait and detect threats against the body, whether that might be a bacterial, fungal, viral threat, and then initiate the corresponding immune response. Dendritic cells are known as antigen-presenting cells, and so they'll take those antigens from that bacteria or virus that they've come in contact with, and then use those antigens to activate and target an attack against those threats in the body.
What's the case for making a cancer vaccine using dendritic cells?
So with a lot of the development within immunotherapy, right, there's a recognition that we can harness the immune system to ultimately attack cancer, right, fight cancer. And that started with checkpoint inhibitors, but has advanced to various cell therapies. When we think about dendritic cells, and some analogies, for the non-PhDs, it's easy to think of them as the general of the immune system. And so if we can get a completely top-down approach targeting a patient's cancer, you could make an argument that that would be a very comprehensive and effective way to do it.
The idea of developing a cancer vaccine built on dendritic cells is not new. Listeners may recall the rise and fall of Dendrion a decade ago. There have been a number of issues surrounding dendritic cancer vaccines. In part, this has to do with a lack of efficacy because of the tumor microenvironment. Can you explain what the challenges have been?
Absolutely. To your point, the lack of efficacy has been the primary knock against dendritic cell vaccines for cancer. They've been widely regarded as safe, and many would make the argument that they're safe because they don't do much. I would maybe potentially take a different approach and say it's more than just the tumor microenvironment that has made prior dendritic cell vaccines fail or be potentially ineffective. The discovery of how we do our dendritic cell vaccine, this novel protocol, I'm sure that we'll be talking about later as part of this double loading, really takes advantage of initiating a Th1 immune response. Prior attempts were not initiating this Th1 immune response because they were singly loading these dendritic cells, and so they weren't being properly activated at the foundation level or at the starting point.
Let's talk about that. Diakonos is developing dendritic cell vaccines that are made with a patient's own immune cells combined with RNA and proteins prepared from a sample of their tumor. How did this approach come about?
The discovery was made in 2006 by a gentleman, Dr. Will Decker, while he was at MD Anderson. And he was working with dendritic cell vaccines and came up with the idea of what would happen if we loaded the dendritic cells on both MHC class 1 as well as MHC class 2. as I was mentioning before, prior attempts really only focused on loading the cancer antigens either on MHC class 1 or class 2, but not simultaneously. And so when he conducted that experiment where he was doing this dual loading, he saw an exponential increase in the T cell activity post the double loading and the interaction with the dendritic cells. And that was the effectively beginning of what Diakonos now is doing in clinical trials.
How does this address the challenges that have been seen with dendritic cell vaccines in the past?
What Dr. Decker discovered was when the dendritic cell has MHC class 1 and class 2 loaded with identical antigens, there's a complex in the dendritic cell that was previously undiscovered. And that complex will compare MHC class 1 and class 2 antigens. And if they are matching, it will recognize that the same threat is potentially directly affecting the dendritic cell as well as also presenting challenges to other cells outside of the dendritic cell, which would be very similar to what happens in a viral infection. And so this previously was not done before. And so to answer your question specifically, how is this addressing those issues? We are effectively mimicking how a virus would infect a dendritic cell. Our bodies are very good at fighting off viral infections, and we have to do it in a very rapid manner because we can't kill the virus. We simply have to destroy the virally infected cells faster than they can infect other cells. And so if we can replicate that process, but instead use the patient's cancer antigens, then we already are taking advantage of a very well established and effective mechanism within the body. And instead of attacking virally infected cells, now the immune system is attacking cancer cells as if they were virally infected cells.
How do you see this fitting into the growing arsenal of immunotherapies, and how do you determine what particular type of cancer it might be best suited for?
Certainly, it's very exciting to see all of the different approaches to immunotherapy, whether that is a cell therapy or a novel compound or an ADC. Where we think this fits in really well is really for the most difficult and challenging cancers, especially cancers that are heterogeneous, like glioblastoma, where single antigen approaches or, selecting a few neoantigens to target may not be sufficient to ultimately eradicating or overcoming the cancer. And so this process is not as easy as an off-the-shelf monoclonal antibody, and we recognize that. And so we think that this is really suited for those difficult to treat cancers, though we would still see and argue potentially even better effects on earlier stage or potentially quote unquote easier to treat cancers that may be a little more immunogenic.
Diakonos is pursuing glioblastoma. For listeners not familiar with it, can you explain what it is?
Yes, glioblastoma is the most aggressive form of brain cancer. It's also the most common and deadly form of malignant tumors in adults. The glial cells ultimately are the result of the glioblastoma, or I should say the mutation of the glial cells in a patient's brain ultimately result in glioblastoma. It has been historically plagued by lack of effective treatments. And so there has not been substantial progress. 5-year survival, especially depending on the type of glioblastoma that a patient is diagnosed with, is anywhere from 5 to 7%, with median survival ranging anywhere from 14 to 18 months.
What treatment options exist today?
So there's a variety of new innovations within glioblastoma, but the primary standard of care is surgical resection if possible, followed by temozolomide and radiotherapy. There is new devices such as tumor treating fields, the Optune device specifically, that have shown some promise in adding additional months to overall survival, though patient use and adoption is relatively low because those devices create pretty significant burden on the patient to use continuously. There are other medications that are in development as well as have been approved, but they appear to really help in potentially extending progression-free survival and not necessarily improving overall survival. So we think there is still a significant unmet medical need for glioblastoma.
And what's the prognosis for someone diagnosed with the condition today?
Right now, it's right around 15 to 16 months. Within glioblastoma, there's two subtypes that really will determine or have a dramatic effect on median survival. A patient that's diagnosed with unmethylated GBM, where the MGMT promoter gene is unmethylated, has a far worse prognosis, with median survival being somewhere around 12.7, 13 months. If you are fortunate enough to be diagnosed with methylated GBM, where that gene is methylated and therefore the cancer is more responsive to temozolomide, median survival is somewhere around 21 months.
Oh, what's your experimental cancer vaccine, DOC 1021, and how does it work?
So the DOC 1021 is based on this double loading principle, but we do add some things into the protocol beyond just double loading that we think really help and differentiate it from prior dendritic cell attempts. So Doc 1021 uses the patient's tumor, right? So after surgical resection is completed, we will take some of that tissue. We'll also apherese the patient to collect the monocytes that we ultimately differentiate into dendritic cells. And then we go through that double loading process that you and I discussed earlier, where we're taking the cancer lysate as well as the cancer mRNA and directly loading those onto MHC class 1 and class 2 respectively. Once the dendritic cell has been activated or educated with the patient's tumor, right, the specific antigens, we'll take those dendritic cells and use an ultrasound guided injection, and we'll make an injection perinodally to the deep cervical lymph node chain. And that allows us to really follow the biology of how dendritic cells would mature after detecting a threat. And so by placing those dendritic cells right next to the lymph node, that is the tumor draining lymph node, they're able to mature and then ultimately activate the corresponding T cells and helper cells to initiate both powerful immune response as well as potentially develop immunological memory against that cancer.
What's known about it from studies that have been conducted to date?
We recently completed our phase 1 study for GBM. We have ongoing studies in pancreatic cancer, and we just opened one for angiosarcoma. But specifically within this phase 1 study that we completed for glioblastoma, we saw a dramatic improvement in 12-month survival as well as overall survival. So within our subset of patients that were majority unmethylated, actually 15 out of 16 of the newly diagnosed patients in the study were unmethylated. 12-month survival under standard of care is expected somewhere around 53%. We saw 89% 12-month survival with a more than a 13-month follow-up. And we have patients that survive significantly longer than that and are continued to be alive. So we've seen statistically significant improvement in overall survival. In addition, it was incredibly safe. We had no attributable SAEs or serious adverse events attributed to Doc 1021. So we have something that seems to be really moving the needle, but also not increasing any risk for treatment.
Some people may be surprised by the enrollment criteria for the study. You took a broader group of participants than most glioblastoma studies. Can you explain the enrollment criteria and why you accepted a broader group of patients than most other studies?
Yes, so from an inclusion criteria, I think it might be even easier to talk about, what we looked at from an exclusion criteria standpoint. And so we had a very broad age range in addition to really not looking for anything beyond significant autoimmune issues as reasons to exclude patients. And so where other phase 1 or phase 2 GBM clinical studies may exclude patients that have a subtotal gross resection or a resection that leaves residual tumor behind, patients that would have progressed or had progressed prior to receiving the treatment, typically those are exclusion criteria, are part of the exclusion criteria. We did not exclude those patients. And so we like to think that we treated some of the most challenging GBM cases. 10 out of 16 of those newly diagnosed patients had either progressed prior to receiving Doc 1021 or had a subtotal gross resection, as I described. And so those patients, if you look at that subset of population within standard of care, typically have a median survival of 7.7 months. And so they typically fare much worse. And so to see the results that we were seeing, not just compared against GBM, but these specific patients, we are incredibly encouraged. As the rationale for doing it, this really credit goes to Dr. Will Decker and in the trial design that he implemented. He really wanted this to be a drug that could benefit the large majority of GBM patients, as opposed to somewhat cherry picking the perfect or ideal patients, which would obviously make that problematic for the BLA filing and the ultimate drug label.
Was there concern about the risk of doing that? Was there internal discussion about the criteria?
Well, the trial was actually started as an investigator-sponsored trial, so there wasn't so much discussion where, maybe you've had some more business-minded folks that would have probably pushed against that in the room. And so he, knowingly took a pretty significant gamble, but I think that was based on all of the work that he had seen preclinically and the success that they had had. And, when you think about the approach, it is somewhat elegant in how straightforward it is and what we're taking advantage of as far as what natural biology allows. So no, I don't think there was significant discussion, though I don't know if anyone would have been successful in dissuading him from doing that. And we're certainly all very happy that he did design it that way, as we now have seen the results.
Well, what's the development path forward?
So right now we're focusing on the phase 2 study. And so with these really exciting results from our phase 1, including efficacy and safety, now we are designing a much larger study to confirm those phase 1 results. So the intention is to develop a 120 patient trial that would be 1-to-1 randomized with a placebo-controlled group. Right now we're talking to a variety of different clinical sites. We've had a lot of interest as the data has gotten out. And so we're intending to start that trial hopefully in early Q1 of 2025. That's what we're on track to do. And with that trial, we hope that we have the opportunity for potential accelerated approval. So it will be powered to generate a statistically significant result, even if we see considerably less efficacy on the DOC-1021 arm than we had seen in the phase 1 study.
And how is Diakonos funded to date, and how far will existing cash take you?
So we have been funded through equity investments through a variety of different investors, both angel investors as well as some institutional groups. We've raised to date just over $17 million. That, as far as our current runway, would take us through the next 24 months. But we are going to be opening a larger Series A round later this year to completely fund the phase 2 trial, as well as initiate some other phase 1 trials in a variety of different indications.
Jay Hartenbach, Chief Operating Officer of Diakonos Oncology. Jay, thanks so much for your time today.
Thanks, Danny. I really enjoyed it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.